prokaryotic chromosome. Prokaryotes usually have one circular chromosome. It bears less morphological resemblances with the eukaryotic chromosome. The genome length of the prokaryote is smallest, particularly in case of RNA viruses. In case of RNA viruses, there can be the number of the genes up to 150. While in case of E. coli, there can be 3000 up to 4000 genes which are organized on the genome of the E. coli. There are 4 million nucleotides of the E. coli chromosome. DNA loops in the E. coli chromosome. There are approximately 50 DNA loops in the chromosome of the E. coli. They are coiled and they are organized in the form of supercoiling. Replication of the prokaryotic DNA. In case of prokaryotic DNA, first the chromosome will have to relax. For relaxing the chromosome, there is an enzyme. The name of that enzyme is DNA gyrase. The function of the DNA gyrase is to remove the helix or the supercoiling which is present in the bacterial DNA. Here is the diagram of the bacterial chromosome. Bacterial chromosome is single, circular in shape, and it is located in the nucleoid region. We can see that this is the coiled and twisted structure of the DNA of the prokaryotic cells. The DNA of the prokaryotic chromosome can be relaxed and it can be in the form of coiling. Here we can see the structures of both. This is a relaxed circular chromosome, while here if we see two turns, if they are removed, the DNA forms a bubble. The both strands of the DNA they separate from each other, while on the other hand, if there are two additional turns, then the DNA goes into over-rotating. Types of the coiling. There are two types of the coiling. One is called as negative coiling, while the other one is called as positive coiling. So both coiling we can see in the diagrams. Mechanism of unfolding of the bacterial chromosome. In this diagram, we can see that the folded supercoiled DNA of the bacterial chromosome, while if these folding have to remove, then there are nicks produced at particular locations. Once the nicks are produced, then DNA is relaxed. Like here is the example. Here a nick is produced on this strand. After producing the nick, the DNA is relaxed here, and in another nick is produced on this strand, on this fold of the chromosome of the bacteria. Once the nick is produced, DNA is relaxed here. We can conclude that prokaryotes have a single circular chromosome that is present within the nucleoid region, and this chromosome has coiling and supercoiling.